Hello. I think that there was some issue with the sound. Uh, as I said before, for those that couldn't hear, I was just saying that today I'm going to be making the presentation of JASCO, making an introduction, and also talking a little bit about the NRS 4000 series, that is the instrument that Dr. Saona used for his research. What I mentioned before when I showed the overview slide is that I'm going to be talking for 10 or 15 minutes, and then Rodriguez Saona is going to talk a little bit about his work in fibromyalgia and using the Raman analysis. And we are going to have at the end Q&A session for 10 to 15 minutes. I just was telling you that if you have any issues, please just let us know if there is no sound, or if you have any issues with looking at the presentation. Also, I just want to let you know that at the end, you can make the question, and you can make the question as well while the presentation is going on. There is no need to wait until the end of the presentation to make your questions. As I was telling, I'm going to be talking about the JASCO Raman products. And what you can see is that JASCO have 4 different instruments for the Raman spectrometers. On the left-hand side, we have the RMP 500 series, that is our portable Raman probe instrument. Then we have our standard model, that is the NRS 4000. And this instrument has it have the capability of the high-speed imaging. It has a focal length of 20 centimeters, and it can hold only one detector, which can be CCD, EMCCD, or also an in-gas detector. It's basically used for quality control environment and academia. And below you have some of the application like microplastic, multi-layer materials, and carbon materials. In the right-hand side, we have the high-end instruments, and these instruments are the 5000 series and the 7000 series. And this instrument has a larger focal length of 30 centimeters and 50. And this instrument, they can hold 2 different detectors. They could have a CCD or in-gas at the same time. And it's preferred for those measurement of high spectral resolution and low wave number measurement. A very specific application that stress on impurity in semiconductors, that you really need to have a very, very small spectral resolution. This is our NRS 4000 series. It's our compact model. Well, the image is self-explanatory. You have the dimensions and the weight of the instrument. Is our compact version of the Raman microscope. Is a very useful for novice users. There is a lot of automation in the software. The lasers, grading, time can be controlled from the software. That means that if somebody is a user wants to change the laser, it can do it from the software. Also, all these combined make it a very high performance instrument. So standard configurations of the instrument is it comes with one laser. It could be 532 or 785. 532 is going to have a 900 grading or the 785 have a combination with the 400 grading. It usually comes with a CCD detector and H filter, but you have the option for E grade or notch filter. Objective lens 5, 20, or 100 for visible range, or NIR if the user decide to go with 1064 laser. Regarding the edge filters, the standard is the edge filter that goes up to 100 wave number, or the E grade that goes to 50 wave number. This in Raman, remember that in Raman it is much more easier to go to this low wave number. It's a different as in the FTIR. As I mentioned before, these instruments have a lot of automation. That means we can do the laser auto alignment. Also, we have Raman light, the Raman light path auto alignment also can be done from the software. And we have the imaging lenses. I'm going to explain a little bit about that later. And what the advantage to use the imaging lenses is that it's going to, no matter which magnification we're going to use, there is no loss of light. But I have one slide for that. As I was mentioning before, we have 532 and 785 as a standard lasers. We have options that go from the blue 405 up to the near IR of 785 and 1064. For the case of the NRS 4000 that is using a 1064, is going to hold only one laser and is going to hold only one detector, that is the InGas detector. That is an instrument that is especially designed for biological samples, or as you are going to see in a few minutes, it can be used for biological or even food industry sample. Resolution, the special resolution that we can achieve is 1 micrometer in X and Y and 1.5 micrometer in the Z. Of course, this is going to depend on the type of laser that you're using, also the objective and the pinhole. We have different pinholes and slits that it goes before the grading, and that is going to determine the amount of light that is going to pass through the grading and then to the detector. Very useful if a user is trying to measure samples with high fluorescence. You can decide to go with the pinhole that it have a smaller area when the Raman signal is going through to the grading and then the detector. Of course, all the instruments from JASCO or the microscope from JASCO has a class 1 laser safety. I recommend that you look into our first Raman webinar. I think Dr. Burgess made a very strong case why this is important for the safety of the users. As I was mentioning before, very ease of use. It's a flexible analysis and reliable. We can look into the laser spot, onto the surface of the sample. It's very easy to switch in between lasers and also gradings. And we have also an accessory that allows us to measure 6 samples, and the microscope is constructing in high stiffness housing, minimizing the drift of the measurement. In the previous slide, I mentioned like the special accessory. This is for deep stage. So it can give you much more space for those tall samples. or thick samples, and at the same time give you the space to place any environmental cells, gas cell, heating, and cryostats. I was mentioning the imaging lenses, and actually this is how other competitors, how they do it, is like high objective lens, high magnification objective lens. They are going to have a different focal point, and the image lens is going to go straight to the monochromator or the mirror before the monochromator. But when a low objective or low magnification lens is used and is changed the focal point, then there is some light that is lost in the mirror. The way that we do in JASCO is we have two different imaging lenses, and actually that is for we can change imaging lenses depending on the magnification of the objective lens, so there is no loss in the light, and of course there is no loss in the Raman signal. As a consequence, we have an instrument with a very high signal to noise ratio. So some of the summary is that, as you can see there, is we have a class 1 laser safety instrument, high signal to noise. Very important, there is a high automatic, it's a highly automated instrument, and it has a lot of features that it helps novice users. The instrument comes with one laser, but it can move, if someone decides to add 2 more lasers, it's possible. A special case, and I want just to mention this to give the opening to Dr. Rodriguez is that we have 1064, and that type of laser, what is recommended is the in-gas detector, which is more efficient for in that range of the infrared. That's the reason why it's not used the CCD detector. Besides that, of course, you are recommended the near IR objective. With that, I would like to pass the presentation to Dr. Rodriguez Saona. I'm going to make him the presenter. So just I want to read a brief introduction to Dr. Saona, and then he can start. Well, I just want to say that Dr. Rodriguez Saona, he's a food engineer, graduated from Universidad Nacional Agraria from La Molina, Lima, Peru. And then he received his master and doctorate degrees in food science from Oregon State University. After he went for a postdoc in the Food and Safety and Applied Nutrition FDA. And then he joined the Department of Food Science and Technology at The Ohio State University, which is currently a professor working on the application of vibrational spectroscopy for chemical detection. Through the collaboration with leading optical sensing industry, his molecular vibrational lab is recognized for the integration of pattern recognition analysis to complex spectral information to screen for potential contamination and adulteration of foods. His research has generated over 90 peer-reviewed articles, 20 books, chapters, and multiple presentations at national and international meetings. Today, he's going, as I mentioned before, he's going to be talking about his research on fibromyalgia and rheumatology disorders using Raman Microscopy analysis of blood spots. With that, Luis, now is your turn. Thank you.
Thank you, Carlos. And I want to thank JASCO for the opportunity to give this presentation. And so we're going to be talking about our research that we have been doing using the Raman microscope in looking for a biomarker for functional disorders in humans. And you may be wondering about the picture that we have in this first slide. It's actually how everything started. We started actually from the interest of a professor in veterinary medicine that worked with cats, and cats that suffer from feline interstitial cystitis, or FIC, which is similar to the syndrome that humans have that is called interstitial cystitis. So we started working in a cat model, and we obtained very exciting results, and that prompted us to look for collaborators in the medical school. So this is a project that have integrated several faculties within the Ohio State University. We have Dr. Tony Buffington. He was the spark plug that kept us going for more than 10 years doing research in this area. Dr. Kevin Hackshaw, he is a rheumatologist with a vast experience in fibromyalgia. Dr. Monica Giusti, that is an expert in LC-MS and metabolomics, and myself, that I have my interest in rapid detection methods, especially associated with vibrational spectroscopy. So we have been collaborating for about more than 10 years, trying to tackle this very interesting topic of disease diagnosis by using these vibrational spectroscopy techniques. So as I mentioned, and also Carlos mentioned before, I am a doctor in food science. So I am not a medical doctor, and so that's why we have expertise in that area. But since I have been involved in this research, I have been picking up a little bit of information. So as you can see, the fibromyalgia is one of several syndromes that are going to fall within the category of functional disorders. In these categories, we have other syndromes such as PTSD, IBS, IC, migraines, that are difficult to diagnose. So the fibromyalgia syndrome is a chronic disorder of unknown cause, and there's large number of individuals in the US that suffer from this disorder, and it has a really high impact, economic impact in our society. It also impacts the quality of life of patients, and FM remains a diagnosis of exclusion. So that means that patients are going to be screened for different diseases that have diagnostic tools. And if they are not diagnosed with this by any of this testing, so they're going to be associated with the fibromyalgia. So there's a need for a reliable biomarker that can detect fibromyalgia so that we can have early diagnosis and treatment. this can positively impact patient quality of life and productivity and release economic resources within our healthcare. So we embarked in this, this research because we felt that it was important that we can, you know, test our hypothesis that we can generate a rapid diagnostic tool for this fibromyalgia. One of the things that it's important to point out is that diagnosis of fibromyalgia can take up to 3 years or more, and it's a long, painful road for the patients. So when we talk about screening methods, now we're going to be going more into my field of analytical chemistry. So what are the different approaches that we have? We can use targeted approaches, and targeted approaches are very sensitive and specific methods that provide good quantitative outcomes, but they rely on one or more marker compounds. So we know the identity of these marker compounds, and we can use sophisticated instrumentation to very accurately screen for these compounds. In the case of fibromyalgia, we don't have such a unique marker compound, or at least it hasn't been reported. So what we relied is on a non-targeted screening approach. Non-targeted approaches, basically we're going to be profiling the signal for the whole sample that we're analyzing. So we're looking for any changes in the composition of that sample that might be different from a control or from other known diseases. So this is a very powerful technique, and you need technology that will provide a wealth of information. So usually the technologies that we rely for non-targeted screening has been NMR, mass spectroscopy, and vibrational spectroscopy. And these technologies have relied in powerful statistic methods that are called chemometrics. So we rely on chemometrics to try to unravel this wealth of information and try to provide some unique diagnostic tool. So when we talk about the omics, as I was mentioning, we have NMR, MS, and vibrational spectroscopy. Vibrational spectroscopy is the area that I have been working for more than 25 years. And I like the technology because it's simple, it's sensitive, specific. So these are key terms for a diagnostic tool. It lends itself very nicely for high throughput analysis. In most cases, we have a sample that is not destroyed during the analysis, so we can use the sample for further investigation, and it has low operational costs. So once you have the system, there is very little investment in running the tests. So what is Raman spectroscopy? So Raman spectroscopy takes advantage of the the scattering properties of molecules. So when a laser with a monochromatic wavelength interacts with molecules, most of that energy can be scattered in the form of radius scattering, which is going to have the same frequency. Now, some of the scattered light will also have a shift in the frequency, and that is what we call the Raman shift, or the Raman event. And this scattering of light is going to be dependent on the type of functional group that is present in the molecule. So, and they are going to be similar to fundamental vibrations that are observed in the mid-infrared region. So, what molecules are going to exhibit this type of Raman shift? And we're going to be looking at molecules that are going to have the ability to change the polarizability of the molecule. So basically, it's going to change the size of the cloud in that molecule. So it distorts that cloud surrounding that molecule. So we're going to see, like we depict here, the ones that are going to show the Raman effect would be the symmetric vibrations for CO2, because now we're changing that cloud, that electronic cloud that is going to be surrounding the molecule. So this is going to be complementary to the information that we get from FTIR. With FTIR, the signal is going to be associated with the ability of a molecule to exhibit a dipole. So usually you're going to see polar molecules that are going to give you a much higher intensity of signal with FTIR. Opposite to that, with Raman, we're going to see better signal from aromatic molecules. So we're going to have that that complementary information. So in general, molecules that absorb really strongly in the mid-infrared are going to be poorly absorbers in Raman, and vice versa. With Raman, we have the ability to collect data very quickly. In most cases, no sample preparation. We can have the laser can go through glass or plastic, so we can actually make measurements through containers. It doesn't see water, so we can use aqueous solutions to probe for Raman. There's no need to collect a reference spectrum. It's used for identification of components. It has excellent capabilities that lend for microscopy, as Carlos already mentioned. And we have easy access to the low frequencies. So as Carlos also mentioned, we can go down to 100 wave numbers and higher. So these are regions that we're not going to be able to collect information with the mid-infrared. So lots of advantages. So you might say, why not everybody is using Raman spectroscopy if it's this excellent technology? Well, everything comes with a price, and the price that we pay with Raman is fluorescence. So Raman can be overwhelmed by fluorescence, and the fluorescence event is going to be enhanced by lasers that are going to be in the UV and visible range. So if you're using the 532 and 785 nanometer lasers, you're going to see the influence of fluorescence. As we show here for acetaminophen, that is the main component in Tylenol, if we use a 532 nanometer laser, we're going to see this hump that is going to be masking some of the signal that from acetaminophen. Now, if we go to a 785, now we can improve the resolution of these bands. But if we go even to an infrared laser, now we avoid that, that fluorescence background. So 1064 really has been a key component in measuring when you have too much fluorescence in your sample. So when we have been working with blood samples, usually blood are going to have a lot of fluorophores that are going to give a lot of that background. So we can use 1064, no problem. But there's also going to be a decrease in the intensity of the signal. So if we are going to be the lasers with lower wavelengths are going to have higher intensity of the signal. If we go to these near-infrared lasers, we're going to have decrease in the signal in the levels of power to the 4. So we're going to have a decreased signal. Now, we're using very sensitive detectors, in-gas detectors, that are going to be able to pick up that, that signal and provide us with good information. So this is some work that we did with extra virgin olive oil. And I just wanted to use this, this slide to show the complementary nature of mid-infrared and Raman. The mid-infrared is in black, the Raman is in the blue, and as you can see, when you see a very high intensity peak for the mid-infrared, the Raman signal is low. When we have very small signal with the mid-infrared, now we have a very good intensity for the Raman. So it usually allows us to look at, you know, different types of responses and try to understand which of these techniques will provide a better outcome for our research. As I mentioned before, we use multivariate analysis when we are looking at the data. So multivariate analysis allow multiple variables to be analyzed at once. We also call them machine learning techniques, and we rely on supervised learning that is going to allow us to discover patterns in the data that relate different attributes with a target attribute. So we're basically going to be looking at multiple variables, and we're going to be associating that signal with a unique attribute or a class. These patterns can be used to predict values of the target in future instances. So by using this machine learning, we're going to develop an algorithm that is going to be predictive. It uses a lot of mathematical analysis, but we rely on very powerful softwares to analyze the spectra. And what is important is to understand what type of output this analysis is giving us. So one of the key things that we're going to be looking in the data that I'm going to be presenting is that the data is going to be surrounded by a probability cloud. That probability cloud is going to basically create a space, a multidimensional space that is going to allow us to predict a class, to identify if a sample, an unknown sample, belongs to a particular class based on that spectral pattern. Now, another advantage of multivariate analysis is that we can use the same information, that same spectral information, and do regression analysis. So we can convert that spectra into a quantitative output. So with the same spectrum, we can do qualitative and also quantitative analysis. So it's very powerful for disease discovery. One of the multivariate analyses that we use a lot in my lab is called SIMCA, or Soft Independent Modeling of Class Analogy. And it basically creates individual PCA models for each class. It's simple, an efficient method for supervised classification. So we basically, each one of these samples that are in different colors, we have assigned a class based on a prior knowledge about that sample. So this is very important when you're developing any of these multivariate analyses, especially if you're developing a supervised classification analysis, is to know what is the class for that sample when you're developing a training model. So in our case, we have relied heavily on Dr. Hacho, that he, based on his expertise, he has been able to provide us samples of well-diagnosed individuals. So that's what we have been using to assign each spectra to a particular class. So as I was mentioning before, if a sample, an unknown sample, is going to be falling within any of these probability clouds that have been developing surrounding the classes, they will be assigned to that particular class. What I really like of SIMCA is that if a sample does not belong to any of the classes that we have in our training model, it will indicate a 0. It will give a 0 as an outcome, which basically says it doesn't belong to any of these classes. So this is very important for us because it will not force a prediction in one of the classes that we have been surveying. Now, there are other types of multivariate analysis approaches that have been reported in the literature. Some of the most common are going to be PLS-DA, or partial least squares discriminant analysis, random forest, supervised vector machines. So those are some of the ones. Also, you're going to have neural networks that are other popular algorithms that we can use for analyzing the data. Now, it takes a lot of preparation in developing these algorithms, these predictive algorithms. So we start by collecting the identifying what are the best conditions for the analysis of the sample. So that will mean using directly the sample or doing some sample preparation. Then we have different pre-processing methods that we can use to improve the ability of the algorithms to see a difference, if there is a difference. We can also use different pre-treatments, and then at the end we're going to have a final algorithm that is going to allow us to make such predictions. So we have been working with Raman Microscopy. So we have been using an infrared laser for the NRS 4500 by JASCO. We have been very happy with the data that we have been collecting with the system. And also it has allowed us to do high throughput analysis by placing samples in positions that we can very quickly analyze. So the way that we have been collecting our sample is we receive blood spot cards from Dr. Hakshaw that have been identified with a code and with the disease. So for example, here we have a patient 039 that has fibromyalgia, and then we collect the sample. We do 2 hole punches in one of the in the card that contains the blood samples. We then dissolve the soluble components of the blood using a buffer, and we're going to then transfer that blood into a microfiltration device. Basically, what we're trying to do is to separate the bigger molecules from the smaller molecules. So we have a cutoff of 30K Daltons, and so once we have done that, we are going to separate the low molecular fraction. So we have this serum that we have separated from the blood. We dry it, and we then obtain our sample that we analyze for the diagnostic tool. So this has taken a long time to develop, but it has provided us the sensitivity and specificity that has allowed to develop this diagnostic tool. So we take the spectra, and the spectra looks like this. So we have unique patterns that are going to be reproducible for the serum that we have collected. This is a fibromyalgia spectra, and we can see unique bands that are associated with different types of molecules. So for example, we see some vibrational modes associated with phenylalanine. We also can see some that are associated with tryptophan. So we can know the source, or we have an idea of what are these types of signals that we're looking with the Raman. So once we collect the spectrum, the really nice thing that the microscope allowed us is to identify a position that is going to give us the best signal. So we can actually look for a place in that spot that is going to give us a good spectral signal. And by looking at this is the data that we have acquired by using the Raman spectra for fibromyalgia patients, which are in purple, RA, and we also had lupus. So we have been able to separate the patients having different diseases by this unique fingerprinting approach of the Raman. So this is what we call a training algorithm. So it was developed by assigning assigning each spectra to known classes. Now, how do we know that we have developed a good algorithm? So for that, we have done a validation step. In this case, we use an external validation set. So we recruited new individuals, we collected the spectra, and this is going to allow us to determine the sensitivity and specificity of our test. So another thing that is important is it gives us information about the misclassification. So we selected between 8 to 10 samples that were new patients, new individuals, and we tested our algorithm. So the one that I'm showing right now, this one on the left, is our training method. And this is the prediction for those 10 individuals based on our analysis. So again, we have a population of 50 FM, about 30 RA, and about 30 SLE that we tested, but the data is very encouraging. Based on this type of analysis, we had 100% sensitivity and specificity for our test. So we're very excited about this data. Another thing that the analysis provide is indication of which bands are responsible for the discrimination of the samples. So this is something that is important because it provides an understanding about the chemistry behind our diagnostic tool. Now, we have also done LC-MS for the serum. So we basically wanted to ask the question, okay, we have this protocol that we have used to isolate the serum. Is that serum going to have the information that allow us to, there's chemicals there that are going to be discriminating. So this is a chromatogram for the different samples. So, and here we have this small region was the most important. So we have, three very big peaks at the beginning, and then we separated the small bands or small peaks in the chromatogram. Here we have an extension of that or enlargement of those peaks, and they were the most important ones. We used that region to do multivariate analysis, and we were again, it showed that we can separate the different individuals based on their disease. It also indicated some of the peaks that were more important for the analysis. So we're actually currently trying to understand and identify what are the identity of these different bands that were separated by LC-MS. So this is very exciting and also corroborates what we have seen with the Raman analysis. As I was mentioning before, we can use the same spectra to develop regression algorithms that we can actually do quantitative. We use the spectra, and we have been able to develop a regression algorithm that is going to allow to predict what is the pain score for the patients. So this is something that is going to be able to be used for, to monitor patients during different types of treatments and get a less subjective measurement of their FIQRs. So to conclude, Raman spectroscopy provides a rapid, accurate, and sensitive tool for disease diagnosis based on unique fingerprinting. It provides versatility, ruggedness, high throughput, and convenience by using these microscopes. And we have actually been awarded an NIH grant to do work, to increase the number of patients. So we're right now looking for a much larger data set, and we're going to be looking also at different machine learning methods so that we can improve the robustness of our predictive algorithm. So we started with, since 2013, we published our first paper, and then in 2019, we had our research showcased by JBC, but there have been other research that supports the ability of these untargeted approaches to diagnose for fibromyalgia. So we're very excited to be part of this field and trying to provide a diagnostic tool for patients. Our research has received a lot of media reports, which we have been very happy. And so I want to thank you for your participation, and if you have any questions, I can try to answer them. Thank you.
Thank you, Luis, for that really impressive presentation. Well, like Luis said, now you can start making questions if you have some.
if you want to also reach to me, my email is rodriguez-saona.1@osu.edu. So you're more than welcome to send me any questions.
So Luis, I'm always, I want to make a question regarding the type of, I think one of the challenges when some researchers are working with chemometric is which algorithm to choose depending on your application. Do you have any insights regarding that?
My insight would be, you know, I would say I'm a minimalist. So I always start with PCA. So start looking at PCA and try to see if there is a natural segregation or distribution or grouping in your data. Okay. And then I would go to, I like, as I was saying, SIMCA. I think that SIMCA provides very useful information. Look at the number of factors that you're using, you know, because one of the things that you want really to avoid when you're developing these models is overfitting your data. So, you know, try to see, look at the number of factors and how much those factors are helping in predicting or in the outcome of the prediction. So, you know, I think that one would be try to be conservative, because my experience is that one of the main factors that are going to result in unreliability in a model is overfitting.
I see. Yeah. Mm-hmm. Yeah, PCA is a very traditional method, I would say. But actually, you mentioned a deep forest, so yeah, that is.
Yeah, and I like also, like, you know, I like to look at the loadings because those are going to tell me which variables are important in the discrimination, because you want to be able to understand the chemistry. One of the really nice tools that you have by working with vibrational spectroscopy is that it's associated with chemical bonds. It's chemical information. So you can actually go back and look at the discriminating power plots, and you're going to be able to you know, get an insight about the chemistry that is associated with the separation of the bands or the separation of the classes. So, you know, it's important to understand, you know, does it make sense, the type of signal that I'm using to discriminate my samples?
Yeah, that makes a lot of sense. Luis, I passed to you a couple of questions. I don't know if you read them or if you want to read them out loud for you. One is say, how do you identify the molecules from Raman spectra?
So there is a lot of information in so different molecules are going to have a unique vibrational mode that is going to be associated with that scattering. So the signal that you're going to get from Raman is going to be unique for a functional group that is present in that molecule. And there is a lot of information in the literature that you can find the type of molecules that will give you a Raman signal at that wave number. So there's tables that you can look. There's research. I usually go and look for lots of different papers that have done work in with Raman. So it's very interesting, you know. There's plenty of work that you can use to identify these types of vibrational modes. You can also do calculations, you know, but I usually rely on published literature, trying to understand what type of signal I'm looking.
There is another question: is what is the most challenging part of having a representative sample of the dry blood spot?
So basically, is getting you know, we started working with finger by pricking fingers and getting blood samples that way. The way that we have done it with Dr. Hackshaw is they collect intravenous blood. So they basically take blood with a syringe, and then they spot the samples. The blood spots are very stable. We have done reproducibility tests on the samples within a patient. But one of the challenges really is we need the blood to be dry. And because if it's not dry, if you overload that the card, it will mold, and basically you will ruin your sample. So it's very important that you develop good protocols to try to prevent overfilling those spots, because then it will remain moist for a longer period of time, and then you can develop mold.
Let's see. There is a question: which software do you use or recommend to do the chemometric analysis? I think that you answered that. You are using Simca, is what you say, Luis?
We're actually using two different softwares. I like Pirouette by Infometrics. I have been working with them for, wow, 20 years. So it's something that comes naturally. It's a very easy to work software. Another one that I use is Eigenvector. So Eigenvector is another one. My former postdoc, Marcel, he loved R. So he would use R to analyze his data. So, you know, the beauty of R is that it's free, but it's a little bit more complex. So, so there's many different alternatives. I think that at the end, what is very important is to understand what is the information, that output that is given you. Look always for residuals plot, because the residuals plot is going to give information about any problems with the data. Look for the leverage. You know, samples that have a really high leverage can skew your analysis. So, I think that at the end, all these softwares are going to give you the tools to make the best type of decisions, but you just need to understand what is all these different types of diagnostic tools that are given in the software.
Okay.
Yeah, I think that, I always tell my students, with great power comes great responsibility, and these statistical softwares are very powerful. So, in many cases, if you don't want to force the data to give you an outcome, because once you start forcing that data, you're going to start modeling noise, and you're not going to get good predictability.
Another question said, like, have you ever tried to use mid-infrared and Raman combined to generate one spectrum for classification models?
Oh, that's a good question. No, I haven't. Actually, I have a Raman microscope and I have an IR microscope. So we usually collect the same sample in the two different systems, but I have not done it. I know that there's groups that are working on that, but that's a great question. I think it would be very interesting.
There is, I think we have time for a last question. It says, well, there is somebody that he would like to reach you to collaborate. I think Dr. Salana already gave his email address, but I'm going to make sure that he gets all the questions and email addresses of the participants. Excellent. And it says, like, if several peaks are mixed together and make the Raman peak broad, how do you unmix the signal from different molecules?
So there's different ways. You can do, I like to do second derivatives. So there's, you can use derivative transformations to resolve overlapped peaks. So that's the best way to tackle that. So I like second derivatives. You can also do a first derivative. The problem with the first derivative is that it's more difficult to look at the loadings plot and get chemical information, because the second derivative is going to match the vibrational modes, but that is not the case with the first derivative transformation.
Okay, I think those are all the questions that came in. Just have my last comment to this. Well, thank you everyone for coming today, sharing this time with us. Just want to invite you again next week for our coming webinar. The topic is related to fluorescence. We have another special guest, and is the Dr. Andrew Jackson. I believe Dr. Hamilton Sherry is going to be talking about the fluorescence instrumentation, and then we have a second part with Dr. Andrew Jacks. Remember to visit our website for tip and tricks posters on Raman, fluorescent, FTIR, CD, and please visit the knowledge base. They have a collection of thousands of papers related to different research topic, and they were using JASCO instrumentation. So with that, thank you very much. Thank you, Luis, for today. Excellent presentation. I really enjoyed it. And see you soon. Okay, thank you.
Thank you.